So we're going to talk about quantification now, but let me just go through okay. Let me just go through a little bit more of the RNA-seq mapping because I know some of you were having trouble wrapping their heads around it. So we're talking about paired end reads and how you can use this probable distance between them. And I know some of you were asking questions about the aligner and the fact that it has to deal with the introns. So I just wanted to remind you all that here, this example is showing a very short intron. It's just a representation. But in reality, you all know that introns can be hundreds of kilobases long. So you can look in any genome browser and see the actual size of introns. And then you can understand how for a mapping algorithm, it is hard if you have a read that is split and 20 bases of it are in this exon, and the remaining 30 bases are 100 kilobases farther away. So the way that these algorithms deal with it, if they are splicing aware, so it's not the same as having a short gap. An aligner such as Bowtie that aligns to the genome is still able to map sequences that are mismatching from the genome by a few bases, or even that have short insertions or deletions. What it is not able to do is split a read 20 bases and 30 bases, hundreds of kilobases apart. So that it will not do. And the reason we use splicing aware aligners is that they, first of all, they know the most likely positions of exons in the genome because you gave it the reference annotation for that genome if you have one. And they try to align the most that they can of one, of a section of a read. And when they can't anymore, they will look for the remaining section either in the whole genome in case that it's a novel splicing junction that's not annotated. But if it is an annotated splicing junction, they already have the positions where it's more likely for that remaining piece of read to be in, okay? So the hash indexing will serve for any base in the genome. It will have every base in the genome, what words, where you can find those words. But it's not the same as the splicing aware. Splicing aware means it's able to handle large gaps, large, like hundreds of kilobases large. And it's still capable of taking that second piece and finding a new position in the genome that could be hundreds of kilobases away that aligns the remaining of your read. Sorry, I hope I didn't confuse you guys further. Let me know if you have more questions. A lot of the questions you guys asked on Slack, I'd be happy to answer during the class too. So just raise your hand or unmute yourself and feel free to ask. All right, so let's talk about quantification now. So there's a few things to consider about the quantification experiment. So now that you have the mapping position, let's go back and think about where the RNA came from to begin with. So you had, let's say, these 4 cells that you took, you made your RNA-seq libraries from. And these cells have different transcripts represented here by different colors. And you have several different molecules for each of those transcripts. They'll be present in different abundances and they'll have different lengths. So what you have to start thinking about in the RNA-seq experiment is that you will recover more reads from transcripts that are longer. So the red transcript will end up being present in more abundance in the output of your sequencing run. And you will also generate more fragments from transcripts that are expressed higher, at a higher level. So what do we want to find with the quantification? We want to find the actual expected relative abundance of a transcript. So what is the expected number of transcripts for a given fragment or a given gene? It is a result, so the number of reads you capture will be influenced by the actual expression level of that transcript and the fraction of your gene in the sample. And it will also be directly proportional to the length of that fragment. So if you have a longer fragment, you're going to get more reads, and also if you have transcripts that are more abundant. And this is the quantity that we're after. We want what is the expected number of fragments that we actually had in the sample to begin with. So to do the quantification, we have to make some assumptions. And these assumptions are that you have a high library complexity. What does that mean? That means that the number of molecules you have in the library is much greater than the number of sequence molecules you actually recovered. So what you see, as you could see from that previous slide, circled were the reads that you captured, but the actual fragments of molecules that were present in your sample are much more abundant. So you only took a snapshot, took a sample of a much larger pool of molecules. And the second assumption is that because the reads are short, then the read length is much smaller than the sequence molecule length, the actual length of the original transcript. So what you're capturing in the experiment is a portion of that original transcript, which you fragmented and then read only a piece of it. So of course, not all the applications satisfy this. This is what we're saying for regular RNA-seq libraries, but if you have microRNA sequencing, then you cannot expect that the read length is much smaller than the sequence molecule length because microRNAs are short. So it has to be dealt with differently. And if you have small input sequencing, for example, single cell sequencing, you can also not assume for a single cell that the number of sequence molecules is smaller than the number of molecules that were present to begin with. So we deal with it in different ways. We have other ways to get around the amplification problem. And I'm not going to go into it now. It's just I wanted you guys to keep in mind that not all the applications will satisfy these two assumptions we're going to talk about. All right, so what are the challenges for the quantification? So like we saw in the genomic alignment and transcriptome alignment as well, there is uncertainty in the read mapping. So the genes have paralogs that share sequence similarities, so it's not necessary that even if a read came from gene A, it doesn't have a match also to the location of gene B in the genome if these genes are related or share, for example, a domain. So it's hard to define a single true location for each short read to have come from. And even if you do have libraries that satisfy those two assumptions we just said, that are more complex than the number of reads you sequenced, and that the reads are shorter than the actual original transcript, you can only measure relative abundance. The key quantity that we're after is how many fragments were sequenced for each transcript. So the data you have is aligned reads, and now you want to count how many fragments you have of each transcript. So what you want is which transcript generated, what amount of transcript generated the observed reads you have. So this should be easy, right? I can just count all the reads that I got for a single gene. Well, not exactly, because like we said, the reads don't uniquely map, and the genes have different isoforms with overlapping exons. Also, sequencing has a 1% error rate, which also already introduces a little bit more uncertainty on this. And the transcripts are not uniformly sequenced. So for example, you have certain biases that are known, for example, for GC content, where a given transcript will not generate the same amount of reads as another transcript with the same length, but a different nucleotide composition.
Can I ask a question
Yes.
before you go on to the next
Yeah, go
slide?
ahead.
Can you comment more about why you only are able to measure relative abundance? Like, why can't you convert these reads to something approximating an absolute number of transcripts? You know, if you
I think
make some assumptions about how efficient your library construction is and what percentage of your library you're actually sequencing, why can't you extrapolate out to something that approximates an absolute number?
right. So you can never, even though you know a lot of the biases that happen, so a lot of the losses that happen during library constructions, and you know, for example, the number of cells that you put into the experiment, but you cannot guarantee an exact approximation. So if you have, you're sequencing from a pool of cells, right? And even on single cell, you have losses during the library preparation, and you have these biases that will cause different transcripts to be captured differently. So you can never go back, just like you can't be sure of which sequence in the genome originated that read, you can never be sure of exactly the amount of RNA you had in your original sample. What you can be sure of, well, more or less, given these problems and taking them into consideration in your quantification, you can always use the measures of other transcripts relative to the one you captured. So that's kind of the information we have to deal with. So you can, it's a relative abundance. You know how much to expect of one transcript given what you saw of all the others, but you can never get the raw numbers. I'll go through the rest of the quantification. If it's not clear, I'll come back later to this point.
No, that was fine. Thank you.
Okay. All right, so we can try to simplify this problem a little bit. So let's start by saying that we know the gene and transcript annotations. So we know all of them. There's no novel isoforms to be discovered. We know everything that should be there. All right, so now let's simplify it even further and say there's only a single isoform that we can obtain per gene. And let's make an even further assumption and say we only have one read to one transcript correspondence, which means a read that we observe means one full transcript in the cell and not multiple reads per transcript, which is how fragments actually generate reads. All right, so we have this theta of G, which is the relative abundance of each gene. And we have lowercase n, which is the number of reads that align gene G. And lastly, we have capital N, which is the sum of all the lowercase n's for each gene. So this would be your sequencing depth, or more specifically, the total number of aligned reads that you got in your library. All right, so in this case, when you simplify it like that, when a success has the success is the probability that you got a read, has the probability of theta, which is the relative abundance of the gene. And so what is the probability of obtaining lowercase n successes in capital N tries, which is the total number of reads you got? So this will follow the binomial distribution. So it can be written as the probability of obtaining lowercase n successes in, given that you have the relative abundance of gene theta, and then that is equal to the binomial coefficient, which in this case is capital N choose lowercase n, multiplied by the probability of n successes, which is theta to the power of n, and that is multiplied by the probability of having capital N minus lowercase n failures, which would be 1 minus theta to the power of n minus n. So this means that our best estimate of the true relative abundance of this gene G is lowercase n over capital N, or the sum of all lowercase n's. So the best guess at the abundance that we can make is just to divide how many reads for that gene we got by the total reads we obtained. So we wish it was that simple. Remember all the assumptions we made on the previous slide and see how we cannot get a true relative abundance even for this gene based on all the problems we have to deal with in this case. All right, but let's go over the quantification steps again that we did. We have sequence reads and we align them to the reference sequence. That can be the species genome or the transcriptome, like we discussed. And we measure the transcript abundance by counting the reads mapped to each transcript. So this is not going to be accurate when you have multiple isoforms or when you have paralogs that share sequence. And then if you want the true transcript abundance, we can solve a maximum likelihood of the observed mapping given the transcript abundance. So to compare across samples these quantities, the counts have to be normalized. And why is that? So first of all, the libraries will have different sequencing depth. You are not going to be able to control, even though you try, so with pooling accurate amounts, you're not going to be able to control that you sequence the exact same amount of each sample. So first of all, you have to control for that. Secondly, the sample composition might be different. Even though, again, you tried to control your input, you might not have gotten exactly the same number of cells you did in experiment 1 compared to experiment 2. So the most standard normalization, which we're not going to go into in this session, we'll leave it for session 6, we're discussing differential expression and normalization, is transforming the counts you got for each gene into transcripts per million units. And I'll go into it later, but basically you should just know that this measure will take into consideration the transcript length and the sequencing depth, because it's going to put the transcripts into a relative abundance form. All right, so what are we normalizing and why are we normalizing? If I did my replicates and I took care of everything to be right, shouldn't I have like a perfectly correlating scatter plot with minor deviations? Sure, it'll look pretty good, but if I put a line across what would actually be an exact replicate, you'll see what the problem is with unnormalized counts. So as you can see from here, without normalization, the quantification would imply that all the genes, almost all the genes, are higher expressed in replicate 2, which is here on the y-axis. So this is why we use normalization. All right, so let's go back to our quantification problem and now revisit our previous assumptions. We started with, again, we're going to keep it simple and say we know all the transcript annotations that should exist. But now, instead of assuming that we have one isoform per gene, let's say we want to use isoforms. We want to know if there's differential expression for different gene isoforms. So one way to deal with it is to define only one isoform per gene. In this example here, I'm showing you a single gene, and that gene has 2 transcript isoforms, one composed of 4 exons shown in dark blue, and one composed of 3 exons shown in light blue. So how do we define one isoform per gene? There are 2 simple methods that used to be very used for this, and they're called the exon union method and the exon intersection method. So based on all the exons that you see for both these transcripts, you can make a super transcript that contains all the exons known for that gene. This transcript doesn't actually exist, but it's a combination of the 2 that do. Or you can use the exon intersection method, which in this case will use only the exons that are shared between the 2 different isoforms that you do know. So there are problem with, so these are two simple ways to deal with the multi-isoform problem, but they both, and they will be convenient for mapping and quantification, but they will give rise to other problems. So the transcript abundance measured by either of these methods will differ from the truth. The union will end up resulting in underestimated expression for genes that have alternative splicing. And the intersection is going to reduce your power for DE analysis, because it's actually going to be getting rid of a lot of reads that could give you information and more power. And lastly, we're going to give up our assumption of one-to-one read to transcript correspondence. So now we actually know that different reads could have come from the same transcript molecule. And lastly, we still rely on the fact that the read fragments are short, and one transcript, because the reason we give up that assumption is because one transcript will generate many fragments. All right, so the problem with these methods is clear. So how can we do a better approach? The better approach that is used by RSIM and several other methods that try to do a transcript quantification is to do this probabilistic method of isoform deconvolution. So the transcript expression method tries to determine exactly how many which transcript did these reads originate from? What is the probability that each read came from transcript 1 or transcript 2? So it involves assigning these reads to a given transcript, and it must capture the assignment uncertainty. So for example, for the light blue and the dark blue reads, because they map to exons that are unique to each isoform, there's no problem there. So you already know where they came from. Using the quantification from these reads, you can also inform your choice for the reads that are mapping to shared exons. So these are the reads that you need to assign to a specific transcript, given the probability that you saw other reads from those transcripts. All right, so your objective is to get the transcript relative abundance, not the gene level, but transcript level. And you don't know where that alignment actually came from. And what you do know is that you have a certain number of fragment alignments. You know the actual transcripts that should be there, and you also know the fragment length distribution. So let's go back to the fragment length distribution idea. So if you remember that we just went through paired end sequencing, and that you have read 1 and read 2 for each sample. So here I'm showing you two pairs of reads. So you have read 1 and read 2. And here for pair 2, you have read 1 and read 2. And they map to different exons of this imaginary transcript. So first you have to perform a computational splicing and compute the insert distance. So you're not going to calculate the distance based on the genomic distance, but you're going to calculate it once this transcript is processed, and because the majority of your mRNA will be mature in your sample. All right, so you have for pair 1, you end up with distance 1, and for pair 2, you end up with distance 2. Once you do that for all of your paired reads, you can estimate this insert size empirical distribution. So you have a range of probable fragment insert size that you actually observed in your mapping. And you do this for every isoform reconstruction. All right, so let's go through that more carefully. You're going to use that for probabilistic read assignment. So for example, here we have a pair of reads. They're mapping to the first exon and the last exon, and you have 3 possible isoforms. And you need to figure out which isoform they actually came from. So for each isoform, you calculate the distance. For green and purple, the distance would be distance 1. And for sorry, for green and brown, it would be distance 1. And for purple, you would have distance 2, a shorter distance, as you can see here, because it doesn't have this middle exon. All right, so according to our distance size distribution, which of these is more likely to have given origin to this pair of reads? So based on this distribution you calculated, you see that it's much more likely that this pair of reads came from form 3. And this is the basic strategy that methods like meso, cufflinks, and RSM rely on. And because of that, it is critical that you have paired end data for this kind of analysis. All right, so we have this method for isoform deconvolution. The parameter that we want to estimate is the relative abundance of the transcript. And the variable that we don't know is the actual fragment alignment source. But what we do know is the number of alignments, the transcripts that actually exist in the sample, and the fragment length distribution. So the probability of a given alignment A to originate from transcript T, given the distance distribution D that we calculated, and the variable that we don't know, the parameter, which is the transcript relative abundance, is going to be equal to the fragment abundance multiplied by the transcript length divided by the sum for every other transcript that that alignment maps to, that read maps to, of the fragment relative abundance multiplied by that transcript length. And that will be multiplied by the probability that that given alignment with the length of L between the two reads is originating from transcript T given the distribution D. So this is a problem that is solvable by an algorithm used by all of these problems called an expectation maximization. And it will try to, what it will try to estimate is what is the transcript relative abundance, which will maximize the likelihood of obtaining the data that you did. So it will try to estimate this parameter we don't know, the relative abundance of the transcript, given the distribution of fragment lengths that are possible and the alignments that you got. All right, so to summarize, I just wanted to show you that the current quantification models are complex. They take a lot more into consideration. It's not just counting which reads align to the genes. And in its simplest form, we can assume that the reads are unequivocally mapped. We only have one origin from them. And then in that case, you could use the binomial for a single gene because the read counts will distribute multinomial for several genes with abundance estimated from the observed counts. But when this assumption breaks, then the multinomial is no longer appropriate. And the more general models that take care of additional information to accommodate the uncertainty of the data will use the base quality score. So it will inform how good an alignment is given a base quality score. It'll use the mappability of the sequence or how many alignments it got all over the genome. It'll use the information of protocol biases even, or if there is a certain trend to get more reads from a certain region of your genes. And it will use sequence biases. For example, are you more likely to get fragments from regions that are GC rich or GC poor? So handling each of these additional variants involves a more complex model, and the reads are assigned probabilistically. So not only to a different isoform of the same gene, but even for a different locus. So genes that share similarity in their sequences will also get a corrected count based on the probability that the read came from each of them. Right, so it look like in the end? You got through all of this and you have your results. So the genes are quantified, and each gene or isoform is going to have a fragment count value. So that can be the expected count, which comes from these probabilistic models such as RSIM, or the raw count, which is just a count of how many reads aligned there with no probabilistic model. And you also are going to have a normalized value, which we'll go into in another session, of TPMs, transcripts per million, or RPKMs, which I'll go into later. So all the samples are quantified in the same way and arranged into a table. For example, we have 22,000 genes that were observed in, for example, 6 samples shown here. And in this case, the table or matrix is going to give you, for each row I, all the expression of a single gene, say MIR301, in all of your samples. And the column J will give you the expression of all genes, all of these and much more, the 22,000, in one sample J, for example, control replicate 1. So the last thing I want to go over before you guys actually do your quantification and get your results is that RNA-seq libraries don't come all in the same shape and form. So we've been talking about libraries that cover the entire transcript, which are the most common, but there's other methods to generate libraries that try to capture the end of the transcript instead of the whole transcript. And the way they can do that is by either fragmenting and selecting a specific portion of the transcript or enzymatically purifying. And the uses of them are, for example, if you want to annotate transcriptional start sites. So the Phantom project tries to annotate the transcription start sites for all genes, and they use the 5' bias libraries a lot for that. Another reason is if you want to annotate 3' UTRs. So these are also cases where the end sequencing libraries will be useful. You can use them if you just care about quantification and expression at the gene level. So let's say for a given project, you don't really want to find new isoforms, you just want to do a quick count of how much each gene is expressed. This is a very good way to do that, because the depth required for these libraries is a lot smaller, since you're sampling only a small portion of the fragment. And another reason to use them is for low quality RNA samples. Those are degraded to begin with, and therefore you would never get a correct coverage of the entire transcript. So using the end sequencing libraries could be very useful for that. And another reason is single cell RNA sequencing. If you work with the data, you know that it's 3' biased, or even some methods are 5' biased nowadays. It's much harder to get high throughput, full coverage for single cells when you want to sample a large number of cells. But for 3' bias libraries, it's actually quite simple. So this is just to illustrate these 3 schemes. You start from poly A selected RNA for all 3 of them, for example, and you get fragmented RNA, of which you can then select either the 5' end based on biotinylation of the cap, or you can select the 3' end based on oligos against the poly A. And you can also sequence the full length, which will capture all of these regions of the transcript. So what does it look like? Since the next part of this session, we'll be talking about visualization. Here is how a coverage data for full-length RNA-seq library would look like, where you have in regions of exons, you have a higher coverage and they distribute across it, the reads distribute across it. But with 5' bias data and 3' bias data, here I'm showing you for a single gene, IL-23A, you would have only a pile of reads in the very 5' region or in the 3' UTR of that gene. And this is what it would look like across all genes in your sample. So because genes have different sizes, we just normalize this by gene length. So instead of using the actual length of genes and bases, we use bins. So you see that the accumulation of reads in the 3' bias libraries happens in the final end of the genes, and the accumulation in exocage data, for example, happens in the 5' end of genes. And I'll leave you guys with some references if you want to know more about methods we have for quantifying RNA-seq and their problems. And if you want to go into the problem of using expectation maximization to do probabilistic read assignment, this is a great paper. It's by the group that developed RSEM, but this is just the reasoning behind that method. And lastly, I'll leave you with this other link of more resources to think about how to construct your RNA-seq libraries based on the purpose that you want to use them for.